Okay, can somebody confirm that you can hear me?
Yeah, we can hear you.
Kiona, are you trying to talk?
I was just saying that we could hear you.
All right, I'm going to assume you guys can hear me because nobody answered, but um, all right, let's begin. Let me share my screen. Can you guys see my screen? Can somebody at least send a message? What do you call it, chat? All right, today is November 15th. And we are actually supposed to be starting chapter 14. I don't think we're there. I think we're on chapter 8. I don't think we finished chapter 8. Let me check here. No, we didn't go there. Did we cover genes and evolution? Anyone? All right, I'm
Yeah.
not hearing anyone, so I'm not sure what's going on. Why is this not showing me? All right, I don't have a marker in this, so I'm going to assume we finished chapter 8. I'm not hearing anyone from anyone, so let's move on, in which case we're supposed to be starting chapter 14. All right, chapter 14, the principles of disease and epidemiology. As I normally do, I give an introductory slide giving you the major goals and a rough outline of what we'll be covering. So know the terms. The terms in this chapter are extremely important. If you know the terms of chapter 14, you will know over 50% of the lesson. So this chapter is a lot of terms. I've only got a few listed here, the major ones: pathology, etiology, and microbial antagonism. But in this chapter, you do need to learn the terms because that's a lot of the chapter. Also understand the relationship between the normal microbiota and its host. Understand the stages of disease development, and understand about nosocomial infections, what they are, and how to control them. Any questions about what we'll be doing? I'm not surprised I didn't get anything, but I would really appreciate it if someone could confirm that you can hear me. There we got a chat here.
Yes, we can hear you.
Thank you for the chats. My volume down? That's why I can't hear you. The speaker was turned off. Don't know why the speaker was turned off. I never turned it off. If someone could talk now, then I could confirm that my system is up and running.
Yep, we can still hear you.
All right, great. So you actually were hearing me before, but I just couldn't hear you. How do I shut down the chat? Hopefully this won't shut down the meeting. Nope, don't want to do that. There we go. Share my screen again. All right. Pathology is the study of disease. Etiology, the study of the cause of disease. For example, the etiological agent of anthrax is Bacillus anthracis. Any questions about that? Pathogenesis, the development of disease. Infection is the colonization of the body by pathogens or the presence of microbes where the microbes are not normally found. For example, all of us have Staphylococcus epidermidis on our skin, but if you were to get Staphylococcus epidermidis growing underneath the skin in the dermis, that would be an infection, and that would be cause of a disease. Any question about that? So infection where microbes are not normally found, or the colonization of the body by a pathogen, a bacteria that is capable of causing disease. Disease is an abnormal state in which the body is not functioning normally. You should understand that when we're talking about microbes, that actually few microorganisms are pathogenic. Only a very few numbers of bacteria can cause disease. Some bacteria are very beneficial to the host. For example, almost all of the normal microbiota is beneficial to the host. And you could say some of them are commensal, meaning not beneficial to us, but doesn't harm us. The normal microbiota and the host have a relationship, and generally there are large communities of microbes found in association with all animals and plants. These communities are known as the normal microbiota, the microbes that permanently colonize a host. So humans, we would speak about the human normal microbiota, but for any other animal like a cat, you could say the normal microbiota of a cat. And there are a few differences. I mean, cats are mammals, so they have a lot of the same bacteria we do, like Staphylococcus epidermidis, for example, and probably Micrococcus luteus, but I don't know for sure about that one. But then there are some others that cats have that humans do not, like Bartonella henselae, which is a normal microbiota of the cat, but when humans get it, it is a pathogen. Any question about any of that? The normal microbiota are only found in certain regions of the body. In disruption of the normal microbiota, or the host balance with the normal microbiota, appears to be associated with disease and impaired immune function. For example, when somebody starts a dose of antibiotics, you might notice that the patient becomes either constipated or gets diarrhea. That is because the antibiotics hits the normal microbiota and disrupts the normal microbiota host balance. And then that's associated with disease. The patient may get constipation or diarrhea. Usually the normal microbiota host balance reestablishes, and then the patient gets better. And hopefully that happens in a day or two for a patient who's taking antibiotics. Any question about any of that? All right, let's talk a little bit more about the normal microbiota. Locations the normal microbiota can be found or shown in this picture. That includes obviously the entire skin, much of the mucous membranes like the eyes, the mouth, the nose, the throat, the gastrointestinal tract, the lower urinary and reproductive systems, meaning the lower urethra on both sexes, the vagina in females. Places where the normal microbiota are not found are internal tissues and organs. Microbes are not normally found in the blood, the cerebrospinal fluid, the muscles, the nervous tissue, and the bone. And if they are found in these places, we call it an infection. Any question about any of that? The upper urinary and reproductive systems are not normally colonized by the human normal microbiota. And so that would be the kidneys and the ureter. They're not normally colonized by the normal microbiota. And it was thought that the bladder did not have a normal microbiota, but at least the human bladder, I'll tell you a little bit about that in a little bit. At least the female human bladder does have a normal microbiota. And this is something that's new, and we'll talk about that in a little bit, meaning this has only been known for, I don't know, 10 or 15 years at most, where there's actually normal microbiota, at least in the female bladder. Any questions about any of that? All right, the normal microbiota differs from a transient microbiota. The normal microbiota is a region that is colonized by the body, and it's always colonized once the patient is born. And usually when a normal microbiota colonizes a host, it will be with the host essentially for their lifetime or close to their lifetime, meaning Staphylococcus epidermidis will be on most of our skin for our entire lifetime. Any question about any of that? That's the normal microbiota. That differs from the transient microbiota, which may be present for days, weeks, or months, but it's not for the duration of the patient's lifespan. An example of a transient microbiota was my aunt had a Staphylococcus aureus infection, and we went to visit her, and she insisted on hugging us. And after she hugged me, I back then used to test my skin about once a term because we had the capability of doing that. And let me think about that. Could we do that in the lab? Yes, we could do it in the lab. We just need some plates of McConkey's plates. Anyways, I had the students do it and I tested my own skin. And then immediately after I hugged my aunt who had the Staphylococcus aureus infection, I came down with Staphylococcus aureus, meaning I got it to grow on the plates. And I said, geez, I'll have to keep an eye on that. I must have picked up Staphylococcus aureus from my aunt. And fortunately, the next time I checked it about 3 months later, I no longer had Staphylococcus aureus on my skin. So it was a transient microbiota for me. And I was pleased that it was a transient microbiota because Staphylococcus aureus is a human pathogen. It didn't cause me any problems, but it caused a skin infection in my aunt. And for me, it was just a transient microbiota. It was present for, I don't know, more than a few days, maybe a few weeks, even maybe a few months. But after a few months, it went away. The normal microbiota probably kicked it off, meaning it overgrew the Staphylococcus aureus, and I lost Staphylococcus aureus on my skin. Any question about any of that? All right. When we're talking about the normal microbiota and the host, you should realize that humans have about 1 × 10⁻³ human body cells in them. And humans have 1 × 10⁻14 bacterial cells in them, mainly as part of their normal microbiota. that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much, because yes, we have about 10 times more bacteria cells than you have human body cells. And that's just the way humans are. Here's some pictures of some normal microbiota bacteria in the surface of the nasal epithelium. That's the orange cocci here. And that's the probably nasal, not hair, but what do you call it? Cilia. Then there's bacteria in the lining of the stomach. There's the lining of the stomach. And there's rods, or maybe, what is that? Not Sarcenia. Spirochetes, either rods or spirochetes, I'm not sure which, in the stomach. And then in bacteria in the intestine, let me blow that up a little. You can see many different bacteria. That's a bacilli, and there's a coccus that could be either a coccus about to divide or a coccobacillus. There's a coccobacillus. And that's probably digested food right there. All right. Recently, or fairly recently, the human microbiota has been found in some recent places where they didn't realize it was found. What was that, 2014? This author discovered that females have a normal microbiota in their bladder. And it was always assumed that when people tested the urine and they got bacteria, that it was from the urethra. But this author went actually and sampled, I guess, urine from the bladder directly and found normal microbiota, at least in the female bladder. And don't ask me about the male bladder. I don't know the answer because this study didn't look at males. It only looked at females. But males have a longer urethra, so it's possible that the males do not have normal microbiota in their bladder. But it's also possible that somebody just needs to look, and back in 2014, no one had looked yet. I don't know. Any question about that? Another place where they found normal microbiota is in mother's milk. It was always assumed that bacteria in mother's milk came from the outside of the nipple, meaning the skin, maybe a little bit of the, what do you call that? The, whatever the vesicle is that milk comes out of. I don't even know what you call that. Someone who has a anatomy and physiology maybe could tell us. But they've looked, and actually mother's milk has bacteria in it while the milk is in the breast. And so that wherever milk is stored in the females, that there's bacteria there. And it's part of the normal microbiota. And then I don't know why they didn't assume that there would be normal microbiota in the placenta, but because the female reproductive system is open to the outside, and we do know that the lower reproductive system is colonized by a normal microbiota. But they discovered in 2014 that the placenta has a normal microbiota. And obviously the placenta is only around when the female is pregnant. And when she has the placenta, it gets colonized by normal microbiota, and it's a unique microbiota that includes some species which are not normally present in the female, the female host. Any question about any of that? All right, so the normal microbiota and the host establish a relationship. and it's generally beneficial both for the normal microbiota and the host. The normal microbiota benefits because it's given a home. In many cases, it's given food, like the normal microbiota in your gut help us digest the food. And when we eat food, the normal microbiota gets its own food from the food in our gut. So the normal microbiota gets a home as well as food. And then the host benefits because, well, the normal microbiota helps the host. It helps us digest our food. It can start microbial antagonism where the normal microbiota can compete with a human pathogen. For example, the normal microbiota can prevent the growth of pathogens. Like, for example, occupying the niche that the pathogen needs to occupy. Like I gave you the example where Staphylococcus aureus, which is a human pathogen, colonized me. If I had enough normal microbiota on my own skin, I might not have gotten Staphylococcus aureus established on my skin because the skin would have been covered with Staphylococcus epidermidis and other members of the normal microbiota, not giving Staphylococcus aureus a niche to begin growing. But for whatever reason, Staphylococcus aureus did get established in me. I don't remember, but maybe I took a shower before visiting my aunt, and so some of my normal microbiota had been washed away, or maybe it just Staphylococcus aureus got established in a small portion in my skin and then grew. It didn't cause disease in me, and like I said, it was a transient microbiota and eventually went away. But the normal microbiota can prevent the establishment of pathogens. It can compete for resources. Like, for example, in your mouth, we have normal microbiota there. If you get a human pathogen, the normal microbiota might eat all the food in the mouth that the pathogen would need. The normal microbiota can also produce acids, making the environment non-hospitable for the human pathogen. For example, on our skin, the normal microbiota makes our skin more acidic. And also in the urethra, the normal microbiota, or the vagina, I guess, the normal microbiota makes the environment more acidic, and that can prevent the establishment of human pathogens. The normal microbiota can also produce bacteriocins. These are proteins that can prevent the growth of certain bacteria, such as Salmonella and Shigella, where E. coli can produce bacteriocins, which prevent the growth of Salmonella and Shigella. A bacteriocin is something similar to an antibiotic. However, it's much more specific. Like when E. coli makes a bacteriocin, it can prevent the growth of Salmonella and Shigella, but it does not prevent the growth of other bacteria in the intestine. For example, all gram-positives would not be affected, and many gram-negatives would not be affected. Obviously, Salmonella and Shigella are gram-negatives, so some gram-negatives are, but the point is bacteriocins are much more narrow in their spectrum of action than an antibiotic. We do have antibiotics which only work on gram-negatives. I'm trying to remember one, and I don't remember one that only works on gram-negatives, but I remember penicillin G only works on gram-positives, okay? And there are bacteriocins which do work on a few gram-positives. And so a bacteriocin would be more narrow than like penicillin G, which works on all gram-positives. Any question about any of that? I always wondered why people weren't working with bacteriocins when a patient had an infection, because it would be a very good, what we call treatment for someone who got, for example, a Salmonella or Shigella infection, to be given an E. coli bacteriocin. And I did read a paper a few years ago where scientists were studying using bacteriocins to treat patients who had an infection. So it is an experimental treatment now, and I've never seen anything further, but it does work when you have the specific infection, like a Salmonella infection. An E. coli bacteriocin will prevent the Salmonella from growing. Any question about any of that? So microbes, the normal microbiota, can act as a microbial antagonism, competing with the human pathogen and preventing the human pathogen from getting established. Probiotics make use of this, where we consume live microbes and they're intended to exert a beneficial effect. Now, usually when we take probiotics, it's not to prevent the growth of a human pathogen, is to exert other beneficial effects, like help us digest our food. But for example, when you do eat yogurt, it can reduce the chance of getting a fungal infection, a fungal bloom, like, what is that called? I'm forgotten the name of the, what a yeast infection is, what species causes a yeast infection. And if you take yogurt with live microbes in it, you can reduce the chance of getting a yeast infection. This is particularly true for women getting a urinary tract or reproductive yeast infection. That eating yogurt help reduce getting that yeast infection. Any question about any of that? All right, the normal microbiota establishes a relationship with the host, and that relationship is a symbiotic relationship. Symbiosis is just where two different organisms or populations of organisms live together. So the host lives in symbiosis with its normal microbiota. The normal microbiota and the host have a specific symbiotic relationship. And the normal microbiota normally forms commensalism or mutualism. These are two different types of symbiosis. Mutualism is where both organisms benefit, where the normal microbiota benefits because it's given a home, and the normal microbiota in our gut are given food to digest. And so they get food, and then they benefit the host, like E. coli in our gut, helps us digest our food. So the host benefits. That is a mutualistic relationship. Commensalism is where the normal microbiota benefits. It's given a home, it may be given food and resources, and it doesn't affect the host. It doesn't cause disease. It doesn't harm or benefit the host. For example, there's a nematode, one in each of our eyeballs, that forms a commensalistic relationship with us. It doesn't cause any harm to us, so it doesn't eat any human cells. My guess is it eats bacteria that fall into the eye, because it has to eat something. It's possible it eats a secretion that the eye produces, but I'm not aware of any secretions other than mucus and well, I guess there's carbohydrates in mucus. Maybe the nematode eats that. Anyways, the host doesn't get any harm or benefit out of it, and the commensalistic organism gets a home, if not more, out of it. A human pathogen, on the other hand, forms a symbiotic relationship with us, meaning the two live together, but it's a parasitic relationship where one organism benefits the pathogen, it's given a home, and it's given resources from the host, and that is at the expense of the host, where the host is damaged and may get disease. Any question about that? So many pathogens form a parasitic relationship with us. When I had the Staphylococcus aureus growing on my skin, it was benefiting. It didn't seem to do me any harm, so that would not be considered a parasitic relationship. It was commensalistic as far as I know, while it was living on me. And then I eventually threw it off, or my normal microbiota threw it off. And then it's no longer symbiosis. Any question about any of that? All right. The normal microbiota can be an opportunistic microorganism. This is where the normal microbiota becomes opportunistic, meaning it does not normally cause disease, at least in a healthy individual, but under certain circumstances, the normal microbiota can cause disease. For example, individuals with a compromised immune system often become infected with an opportunistic pathogen, like a patient who is an AIDS patient. They have no immune system, and then the normal microbiota can grow in regions where it should not, causing an infection. Some examples of an opportunistic pathogen: E. coli is normally found in the gut, but if it's growing elsewhere in the body, it will cause disease, such as causing an ear infection or causing a lung infection that we call pneumonia. Staphylococcus aureus is found in about 40% of Americans in their nose and throat, and that would be the upper throat. And it doesn't normally cause disease in this region, but if it's growing on the skin, it can cause skin disease. And obviously, if it's growing in the lungs, it causes pneumonia. So an opportunistic organism can cause disease if it's colonizing an area where it's not normally found, like E. coli in the ear or in the lungs. Or let me think about that. Or if the patient is not healthy, and then the opportunistic microbe can grow beyond what it should, like in an AIDS patient, when a normal microbiota of their skin causes skin disease. And I can't think of one off the top of my head. I don't know if Staphylococcus epidermidis could do that, but in an AIDS patient, it might. No, that's not it. I used to know a bacteria that was part of the normal microbiota that caused a skin infection in an AIDS patient, but it slipped my mind what that was. So, any question about any of this? All right, Koch's postulates. In talking about the etiology of infectious diseases, it's important to revisit Koch's postulate. And I think this is the second time we're going to talk about Koch's postulates. We will talk about Koch's postulates 3 times in this class. Koch's postulates were something that Dr. Koch postulated when he realized that Bacillus anthracis causes anthrax, and then he used Koch's postulate to prove that Bacillus anthracis causes anthrax. So Koch's postulate is used to prove the cause of an infectious disease. Let me blow this up. So what you do with Koch's postulate is you isolate the pathogen from a sick creature that may be causing the disease. You grow it in lab, obtain a pure culture of it, and that's not shown here because to get a pure culture, you'd probably grow it up in colonies and then streak it out on another plate to get a second isolated colony, and then grow that up to get the pure culture. And assuming that that's the pure culture causing the disease, you then inoculate a healthy animal, in this case, a healthy bunny, with the pure culture that we think may cause the disease in this bunny. All right. If the bunny gets sick, why the bunny's turned on its side, oh, the poor bunny, he's sick. We then can say, yeah, it looks like this organism caused the disease. Koch's postulate states, though, that you have to go further. You have to prove that the organism that you injected into this bunny and then caused disease can be isolated out of this bunny. And in every case, the sickness is caused by this organism. And that's what Koch's postulate is. Any questions about Koch's postulates? The steps you use to determine if an organism causes a disease. And if you remember, was it Klebsiella pneumoniae? There was a bacteria that, not pneumonia, Haemophilus influenzae is a bacteria that Louis Pasteur isolated from a flu patient, and he assumed that it was causing the flu. In reality, Haemophilus influenzae does not cause the flu. The flu is caused by a virus, not a bacteria. That was misnamed because Louis Pasteur did not use Koch's postulates to show that that bacteria caused the flu. Any question about any of that? Since Koch came up with the postulates, we've used Koch's postulate to show or prove that given bacteria cause a given disease. But there are some exceptions to Koch's postulate. Like, what do you do if the microbe has unique culture requirements and cannot be grown in vitro? Bartonella henselae, which causes cat scratch fever, and then, if I can remember the name of this organism, the bacteria that causes syphilis, which is escaping me, I don't remember the name of that. These organisms cannot be grown in vitro. You can't grow it on a petri dish. And people have been trying to grow these human pathogens for over a century now in tissue culture, and we've never figured out what they need to grow. So we've never succeeded in getting them to grow. So how do we know that they cause the diseases we think they cause, like Bartonella henselae causes cat scratch fever, and the bacteria that causes syphilis causes the disease we call syphilis? How do we know that? Any questions? Any guesses? Nobody has a
Don't,
guess?
don't we use specific type of media?
No, because these organisms can't grow in tissue culture. So if you can't get it to grow in tissue culture, you can't run Cox postulates because you have to grow it in tissue culture to get Cox postulates to work. So what do? How do we know Bartonella and Sally causes cat scratch fever? How do we know that HIV causes AIDS? This one we might be able to grow in tissue culture. Yeah, we can grow HIV in tissue culture, and we could inject it into patients and see if it causes AIDS. We've never done that for moral and ethical reasons. So we've never proven with using Cox postulate that HIV causes AIDS. Because you wouldn't want to inject HIV into a patient. It could give them AIDS and then kill them. All right. So how do we know? Nobody
Do you
has a
just-
guess.
Do you just study the infected individuals?
Yeah, what you do is you study large numbers of patients or people and then follow that group over time. They've done that for HIV, for example, and found some people who are HIV negative became HIV positive. And then you follow those HIV positive patients over time, and they came down with AIDS. So you do large epidemiological studies to show that a given microbe can cause a given disease when you can't use Cox postulates. What do you do if multiple organisms can cause the same disease signs and symptoms? For example, there are many viruses that cause the human cold. So Cox postulate won't work because it's assuming that one organism causes the disease.
Could you do a treatment and see if it responds to known treatments of certain organisms?
You could. However, you have to still say that this given organism causes the given disease. And they've done this. They've actually taken a species of virus and given it to volunteers, and they came down with the human cold. And not everybody came down with the human cold, but everybody who had the symptoms of a cold had the virus in them. So we have done Cox postulate in different ways to show that the different viruses which cause the human cold were responsible for the human cold. Okay. And then you can do epidemiological studies too, like coronavirus. I don't think they've studied that. There's many coronaviruses, even more than one species of coronavirus that cause the human colds. And they've done just large epidemiological studies, and they found people became, let's say, coronavirus number 11. They followed it, people who were negative for that virus, and then they followed them, and they're coronavirus 11 positive, and then some of them came down with the human cold. Okay. And then they've shown that coronavirus, you might not know this, but coronavirus actually was known to scientists before COVID-19 came about. And most of the coronaviruses that were known caused the human colds. And we'll talk a little bit more about that later in the term. And obviously, there is one coronavirus which causes COVID-19, which is a specific disease characteristic in humans, including death. All right, is there anything else I want to talk about that? No. So there are some exceptions to Koch's postulate, microbes that we can't grow in culture. We can't use Koch's postulate. Microbes where multiple organisms cause the same disease signs and symptoms, where there's many different viruses. In fact, there's 3 families of viruses, not just 3 different species. There are 3 different families of viral species which cause human colds. And then moral and ethical conditions where we wouldn't give the human HIV, for example, because it would be moral and ethical reasons not to do that. All right, let's go on with a few more terms. A symptom is a change in a body function that can be experienced by a person as a result of a disease, but it is subjective. It's not something apparent to any observer. It's not something that can be measured. Like an example of a symptom is when a patient gets a headache. Nobody else other than the patient can determine that that patient has the headache because we can't measure a headache. Nobody else can prove that somebody else has a headache. So a headache is a symptom. A sign is a change in the body that can be measured or observed as a result of disease. So this is an objective sign, an objective measurement of disease. For example, when a patient has a temperature of 38.5 degrees Celsius, which that's somewhere over 100 degrees Fahrenheit, we can easily measure a sign by measuring the patient's temperature with a thermometer. So a sign is something that can be measured. A symptom cannot be measured. Now, most of the time when you talk to a patient, and even most clinicians do not make the distinction between a sign or a symptom. Textbooks do make the distinction, and so you should know the definition of what a sign is and what a symptom is. And generally, I will use them more or less interchangeably in the future, okay? Meaning when I say a sign, I mean a symptom, and symptom, a sign, and that's the way most clinicians use them. But textbook authors do say they are different meanings. And it is important, a sign can be measured, a symptom cannot. A syndrome is a specific group of signs and symptoms that characterize a disease. For example, the signs and symptoms that are characteristic of AIDS. A communicable disease, a disease that a patient gets that can spread from one host to another. So example, measles and TB and chickenpox are all communicable diseases. A contagious disease is a disease that easily spreads from one patient to another. So what's an example of a contagious disease? What's an example of a contagious disease? A disease that easily spreads from one patient to another. This is an easy one, people.
Isn't it COVID?
Yeah,
19?
COVID-19, the human cold, flus, measles, these are all contagious diseases. Now, this question is much more difficult. What's an example of a communicable disease, one that can spread, but it doesn't spread easily? I didn't expect many people to know. I just thought maybe somebody might know of a disease that is communicable, but it's not contagious. It doesn't spread easily. There's actually one right here. Another example would be Oh, what's the flesh rotting disease? Leprosy. Leprosy does not easily spread from one patient to another. So it is communicable, meaning that it can spread, but it is not contagious because it doesn't spread easily. The one mentioned here is TB. It is communicable. It does spread from one patient to another, but it is not contagious because it does not spread easily. There are also non-communicable diseases, diseases that do not normally spread from one host to another. Example, tetanus and botulism are two non-communicable diseases. They do not normally spread from one host to another. Can anyone think of an example where a patient can actually get tetanus from another patient who has tetanus? There's only one case I know of.
If a needle was reused?
Yeah, that's actually a good one. You could get a non-communicable disease from a contaminated needle. That is true. For example, a patient who had tetanus, you'd have to stab the patient where there would be tetanus, and I don't know where tetanus is. Obviously, it grows in the nerve system. So if you're to stab like into the brain or something, and then poke yourself, you might get it. The example I was trying to think of is people who are cannibals can actually get diseases that most of us would not get, like tetanus or botulism. Cannibals can get those diseases if they were to eat a patient who had tetanus or botulism. And the rest of us would not get it normally from the patient. Although you're right, with a needle prick, we could get tetanus or botulism. So that was a good example. All right, let's talk about the occurrence of disease. To understand the full scope of a disease, we need details about its occurrence, about the old cases and the new cases. The incidence is the fraction of a population that contracts a disease during a specific period of time. So the incidence is the new cases only. Trying to think, usually when we talk about the flu, they're talking about the incidence of the flu, like the occurrence of the flu in the United States. The incidence is increasing. There are becoming more and more cases, new cases of the flu, and that's because the weather is getting colder and the flu does well in the late fall, the winter, and early spring. So the incidence of the flu is increasing. The prevalence, on the other hand, is the fraction of a population having a specific disease at a given time. And that would be the old and the new cases. For example, the prevalence of AIDS in the United States in 2007 was 1,185,000. But the incidence of AIDS in the United States in 2007 was only 56,300, meaning patients with AIDS, the total number were over 1 million in 2007, but the new cases were only 56,300. Any question about any of that? Usually when we're talking about COVID-19, they're giving you the prevalence number. And I think when they're talking about the prevalence number of COVID-19, we're talking about the patients who had COVID-19, and they may not have it any further, meaning you had COVID-19 in the past, and so you're the number. Now, what happens when somebody gets COVID-19 more than once? And I don't know the answer to that. All right. When we're talking about the occurrence of disease, it's important to look at the disease time period. A sporadic disease is a disease that occurs only occasionally in a population. Most people say that typhoid fever in the United States is a sporadic disease. It occurs occasionally, but not all the time. And usually the typhoid fever in the United States is low numbers. An endemic disease is a disease that is constantly present in the population. For example, the common human cold is an endemic disease in the human population because we always have the cold. It's less common in the summer. It's much more common in the cool winter, but it's endemic to the human population because we always have the common cold. Malaria is a disease we say is endemic to the tropics because malaria spreads in the tropics. And that's because that's where the parasite is found. And then the parasite is spread by the mosquito, and the species of mosquito that spread it are in the tropics or the semi-tropics. An epidemic disease is a disease acquired by many hosts in a short given time. An epidemic is in one region, usually one continent, where the disease is usually acquired by many hosts, and this definition doesn't have it that way, but they say significantly higher than normal. Like right now, we're in an epidemic of COVID-19 in the United States because COVID-19 a few years ago was zero in the human population. And so they say an epidemic in the United States, and initially was an epidemic in China, so in Asia, and then it spread to more than one continent. If it spreads to more than one continent, becomes worldwide, we no longer call it an epidemic, we call it a pandemic. So COVID-19 is currently in a pandemic, a worldwide occurrence of an epidemic. Any question about any of that? So when COVID-19 was only in Asia, we said it was an epidemic of COVID-19 in Asia. And once it spread beyond Asia, it became a pandemic. Sometimes when you have a disease which is very low in number, you can get an argument about what it is. Like, is it tuberculosis? Yeah, tuberculosis in the United States occurs in a very low number in the United States, or low enough numbers. And there's an argument about some people calling it a sporadic disease in the United States. Other people say, no, it's an endemic disease in the United States. And when you get a disease which is at low numbers, the real distinction between a sporadic disease and an endemic disease becomes blurred. And we won't get into that argument whether that is a sporadic disease or an endemic disease. When we're talking about disease, it's important to discuss the severity or the duration of a disease. Another useful way of defining a disease or a scope of a disease is in terms of its severity or duration. An acute disease is a disease which is short. where the symptoms develop generally rapidly, the disease is short-lived. An example, the best example of acute disease is the human cold. The symptoms are fairly mild, they do develop rapidly, but the disease is always short-lived. I've never heard of even an AIDS patient having a long-term human cold, although certainly that would be possible. Another example would be the flu. Most of us don't consider the flu an acute disease because the symptoms are not very mild, but they are short-lived. The disease develops fairly rapidly, and even the worst patient usually gets over the flu after a month or two. A chronic disease, on the other hand, is a disease that develops slowly and persists for long periods of time. An example of a chronic disease would be mononucleosis, where the patient initially will have mild signs and symptoms, and they will get worse. The disease slowly develops, and it persists for a long period of time. And I'm not an expert on mononucleosis, but it lasts for several months. Another chronic disease would be a tennis elbow, where the tennis player would be playing tennis and they damage their elbow, actually wear it out. And as long as they continue playing tennis, they will continue having a chronic disease, tennis elbow. So the disease develops slowly, gets worse, and then persists for a long period of time. Another disease is a subacute disease. We're not going to discuss this, and you don't need to know it, but there are diseases where the patient has symptoms between an acute disease and a chronic disease. An example being subacute sclerosing panencephalitis, where the disease symptoms are between that of an acute and a chronic disease. Any question about any of that? When we're discussing a disease, we should realize that the host can play a role in the development of the disease. So let's talk about the extent of the host involvement in a disease. Infections can be classified according to the extent to which the host body is affected. So if the disease stays in one area, we usually call it a local infection, where the pathogens are limited to a small area. For example, a boil caused by Staphylococcus aureus is a local infection. Only the skin is affected. In fact, only a small region of the skin where the boil is. Abscesses, another example of a local infection. A local infection, in contrast with a systemic infection, and this is a generalized infection, where the infection tends to spread throughout the body. An example of a systemic infection is when a patient comes down with measles. The measles virus goes throughout the body and infects many different cells of the body. A systemic infection. You can have a focal infection where the systemic infection, a generalized body-wide infection, began as a local infection. For example, if Staphylococcus aureus causes a boil, and this boil were to get worse, it can invade the bloodstream, and then the blood can spread the Staphylococcus aureus throughout the body, causing a systemic infection. That would be a focal infection, one that started as a local infection and then became systemic. MRSA can start from the skin, such as a boil, and then be spread by the blood, becoming a systemic infection. MRSA is very similar to Staphylococcus aureus. MRSA is Staphylococcus aureus, but it is multi-drug resistant Staphylococcus aureus. Any question about any of that? Bacteremia is the presence of bacteria in the blood. You can compare and contrast that to septicemia, where we actually have growth of bacteria in the blood. Now, usually when a clinician notices that a patient has bacteria in the blood, they're only observing it. They don't, in which case they call it bacteremia. They don't know if the bacteria is growing in the blood, and that would be septicemia. But if you have growth of blood, bacteria in the blood, it's septicemia. And I don't know how they do that. They must sample the blood and find it growing in the blood. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. Any question about any of these terms? More on the extent of the host involvement. The state of the host can determine the extent of an infection. A host generally can resist an infection, at least to a degree. And you have to break down the host resistance before the pathogen can cause disease. A primary infection is an acute infection that causes the initial illness. So a child has a throat infection and it causes an ear infection. That's the primary infection. A secondary infection is where an opportunistic infection then occurs after the primary infection has weakened the host defenses. And then the opportunistic infection can cause further disease. The child gets an earache from another organism growing in the ear, and that would be the secondary infection. Any question about that? And then there's a subclinical disease, also called an asymptomatic disease, where the patient has the disease-causing organisms, but the patient has no noticeable signs or symptoms of disease. So the patient has an inapparent infection. For example, some people can carry the polio virus, but they never get any signs or symptoms of polio. That would be a subclinical or asymptomatic infection. Most of you are fairly young. Young patients or young people can get infected by COVID-19 and get a subclinical infection where the patient has no noticeable signs or symptoms. And that we call an asymptomatic disease. These people can spread COVID-19 to others, but they themselves never have any signs or symptoms of COVID-19. And that is actually how COVID-19 came into this country. If you remember, Trump barred anyone from coming into the country from China, and then later, I think, from Asia. And that was trying to keep COVID-19 from coming into the country. However, it came into the country by subclinical patients. Somebody brought it in and they didn't know they had the virus. And of young people, I guess I was starting to say that and I don't think I did. Young people, about 50% of you will get a subclinical disease by COVID-19, meaning you will not know that you have COVID-19, but you can spread it to others. And that would be a reason why you don't want to get COVID-19, because even though you will not get, maybe 50% of you will not get any signs or symptoms, you can spread it to other people, like your grandparents, for example, who, if they get it, could die from it. Any question about any of this? Any question about the extent of host involvement in disease? All right, let's go back to the severity or duration of the disease and talk about a latent disease. A latent disease is a disease where the patient has no signs or symptoms of disease. In the case of a latent disease, this is different from an asymptomatic disease or a subclinical disease. A latent disease is where the pathogen is inactive. You cannot spread a latent disease while the disease is in latency, because the pathogen is inactive. The best example of a latent disease is when a child comes down with chickenpox. The virus is actively replicating and can spread from patient to patient. And then the virus goes latent, where the disease organism is present in the child, but the child has no longer any signs or symptoms of disease. And in fact, the virus is latent, will not cause disease, it's inactive, and it's just sitting around. And it can actually sit around for decades and then come out later when the child is now an elderly patient, and the virus can activate and then cause disease, which would be the shingles. And when the patient has the shingles, they can spread the virus. Any questions about that? So anyone who has had chickenpox, you probably do have the virus in you, but it's latent, not causing disease. And at some point in the future, usually when we're elderly, it can come out of latency and cause shingles. Herd immunity is when many people in the population are immune to the disease-causing organism, and then the disease-causing organism cannot spread very much. Let me blow this up. So if the population is not immune to the disease and somebody in the population gets the disease or the disease-causing organism, almost everybody in the population can come down with the disease because there is no immunity to the disease. And then there's just a few lucky individuals which did not obtain the disease-causing organism, and most of the rest of the population will come down with disease. Initially, when COVID-19 hit the human population, nobody had immunity to it, and so they were afraid that this would happen. Large numbers of patients would come down with the disease, and with COVID-19, large numbers of patients would die. And now we have the vaccine, and enough people have had COVID-19 that there are some people who are at least partially immune to it. And so this will not happen because there are enough people who are immune that this won't happen. But with COVID-19, we don't have enough people immunized to prevent the spread of COVID-19. But at least this won't happen because there are a lot of people who are now immunized, either from the vaccine or from getting COVID-19. That doesn't mean you are protected just because you had COVID-19 or even the vaccine. And we'll talk more about that with COVID-19. The trouble is the new strain changes. And so even though you once were immune to COVID-19, you may not be immune anymore because the virus has changed. And we'll talk a little bit more about that later in the term. If, on the other hand, enough people are immunized and somebody becomes diseased, has the disease-causing organism, only people who are not immunized and in close contact with the individual will become infected. Other people who are not in close contact with the disease-causing patient will not become infected, because when the disease tries to infect like this patient or this patient, they're immunized, so they don't get the disease and they can't bring it to this patient. And that's the whole point of immunization, to get enough people immunized in the population to prevent the spread of disease. And if you get enough people immunized in the patient, we call that herd immunity, where there are enough immune people in the population that the disease does not spread, at least not spread much. Unfortunately, with COVID-19, we need 85% or more of the population immunized before COVID-19 can spread. And unfortunately, we are not at that number yet. So COVID-19 will continue to spread. And of course, because the virus changes, somebody who was once immune will have reduced immunity or no immunity. And so they can get a new virus. Any question about any of that? Any question about herd immunity or latent disease? Not hearing anything, so let's move on. I think I'm going to stop here because it's close to 6:20. Any questions on anything so far? If not, I'll see you in the lab at 6:30. Bye.